Hi y'all, I'm Jedidiah, and today I'm going to teach you about the cell cycle. Let's get this rodeo on the road. I'm wearing my interphase t-shirt. You see, the cell is usually in interphase. You see, there's 3 stages of interphase. There's G1 phase, and that's where the cell is just taking a nap and it's just resting. And also taking in vital nutrients like water and antioxidants. Splash cooler. The cell is also taking in good food and good glucose like marshmallows. I can feel my cells going right now. Then goes into the S phase. During the S phase, the DNA starts replicating. Well, Jedidiah, why would the cell's DNA need to replicate? That's a good question, and I will answer that question not right now. After that, it goes into G2. During G2, protein synthesis stops occurring, and microtubules stop forming for mitosis. You see, after interphase, the cell goes into prophase, where the earlier DNA called chromatin turns into chromosomes. Chromosomes are DNA formed of sister chromatids attached at the centromere. As you can see, there is a nuclear envelope around the chromatin in this one, but in prophase that nuclear envelope has broken down, giving the chromosomes room to dance about and get a little square dancing on. That's what we do when we're free and don't have no nuclear envelopes surrounding us. You see, now we're in metaphase, and all the chromosomes are lined up like they're about to do a line dance. You see, we've gone from square dancing to line dancing. And you can also see these right here. I call these the centrioles, and so do scientists. And they're making spindle fibers, and you'll see what those do once we get into the next segment. Well, it's the next segment, and we get to learn about the spindle fibers. You see, they used to be X's, and now we call this the letter V. And these right here are the sister chromatids. And these spindle fibers attached to the centrioles are pulling them apart. What are they doing that for, you may ask? Well, you're going to find out when we go into telophase. Okay. You see, now we're in telophase, and the centrioles have pulled all the DNA sister chromatids together, and then they melted back into chromatin, and that nuclear envelope that broke down to let them do a little square dance is now back on. And you can see, it looks almost as if there's 2 cells inside one, and I don't know how that works, but that's what it looks like. But as you can also see, it appears that 2 cells are about to form, and we're going to find out how that happens when we go into cytokinesis. Now, microtubules produced in the interphase cycle G2 are using to make the cytoplasm and the cell membrane split, and they're going to form into 2 cells. And you see, this is the original parent cell, and this is what we call the daughter cell, even though it's not really the daughter, because there's no mother cell, you see. You know, my mama always said there's like a cell cycle, and always know there's going to be 2 daughter cells. And that, my friend, is the cell cycle, and you would be surprised how much this goes on. And you see, the DNA is going from chromatin to sister chromatids to chromosomes, and you may be asking, you can leave now, thank you for your help. You may be asking, well, Jedidiah, how do you know when to do these things? Well, you know what I say, like a good boot needs a shining, a good cell needs a cyclin. Cyclin is a chemical regulator that helps feed checkpoints for the cell in the cell cycle. And that, my good friend on the other side of the little screeny thing, that is how the cell cycle works. So I hope you'll help me next time as we go look at something else having to do with biology, and keep it real on the other side, my peeps.